to look at the physiology of the female reproductive system, and it was always assumed that females have their total supply of eggs determined at birth, but there is new evidence that suggests that there are stem cells that can arise from the germinal epithelium at the ovary surface and can create new egg cells. But even though that might overturn the previous assumption, for right now we're going to assume females have their total supply of eggs determined at birth. The process of meiosis in females is called oogenesis, and this is a process that produces female gametes. The female gamete is an ovum or egg. Now, oogenesis takes years to complete. It's not as quick as spermatogenesis. Oogenesis begins in the fetal period. Stem cells that are diploid, called oogonia, multiply by mitosis and store nutrients. During the fetal period, these stem cells are also able to differentiate into primary oocytes, or we could say they begin the process of meiosis by entering meiosis 1. Primary oocytes are generally covered by flat cells called follicle cells, and the follicle cells covering the primary oocyte create primordial follicles. But that's as far as the process goes before birth. So these primary oocytes begin meiosis, but they stall in prophase 1. Then at birth, again, remember we're going to presume that females have their lifetime supply of primary oocytes. It's not till after birth that the oocytes that stalled in prophase 1 resume meiosis 1. So after puberty, a few of the primary oocytes are selected or activated, and it's the selected oocyte that is going to be able to resume meiosis 1 on a particular month. And the result is going to be the completion of meiosis 1 and the creation of 2 cells. One of the cells is going to be a secondary oocyte. It'll be haploid. It'll be larger than the other cell, which is the first polar body, a small cell that actually will degenerate oftentimes before it even divides to finish out meiosis 2. But the secondary oocyte will have all the cytoplasm and the organelles of the original cell. And you may or may not be picking up on a trend here that oogenesis is going to basically take a bet on one cell. It first took a bet per month on one primary oocyte, activating it. Now it's going to take a bet on one secondary oocyte. Even though it could create 2 smaller secondary oocytes, it gives all its cytoplasm and organelles to one. It is that chosen secondary oocyte that arrests in metaphase 2. So it got made at the end of meiosis 1. It got to enter into meiosis 2, but it gets no further than metaphase 2 unless it becomes fertilized. So if that secondary oocyte is penetrated by sperm, then it gets to complete meiosis 2. If it's not, it deteriorates. If penetrated by sperm, the secondary oocyte does complete meiosis 2, and it would yield a second polar body, again, something really small, a small cell that's going to generate, and then a large ovum, a functional gamete, an egg. So let's look at these events of oogenesis with this diagram. On the left, we have the meiotic events. On the right, we have the follicular development in the ovary. We can see on the left that before birth, there's mitosis and the beginning of meiosis 1. So the stem cell, the oogonium, becomes a primary oocyte, and then we'll see the arrestment of meiosis 1 during prophase 1 before birth, and it remains the same after birth. And then during infancy and childhood, the ovary is going to be functionally inactive, but it does contain these primary oocytes arrested in prophase 1. It's not till puberty, so from puberty to menopause, that these primary oocytes continue to develop. They'll get to complete meiosis 1 and become a secondary oocyte. Then the secondary oocyte will be ovulated. The secondary oocyte will arrest in metaphase 2 and only be able to complete meiosis 2 if penetrated by a sperm. And if penetrated by a sperm or fertilized, it gets to become a complete ovum. So our oogonia is a stem cell. Our primary oocyte is diploid. Our secondary oocyte is haploid. And then the ovum is also going to be haploid. And we actually create only one egg or one ovum at the end of the meiotic events for oogenesis and 3 polar bodies. But in total, we have 4 cells. Now keep in mind the way oogenesis works, we put all our bets on one cell, and that's when we get only one ovum. And this ovum is going to be much bigger than the polar bodies. Now, we also want to look at the right. We want to look at this follicular development in the ovary. Now, the primordial follicle which is present at birth, is just a primary oocyte surrounded by a layer of flat cells, or follicle cells. Now, we're going to see that the follicle was able to grow with the oocyte at puberty as that primary oocyte gets to complete meiosis 1 and then enter into meiosis 2. So our primordial follicle is going to develop into a primary follicle each month at puberty, the one that's chosen. A primary follicle has one layer of follicular cells that are more cuboidal than flat. And then we go from primary follicle to secondary follicle. So now we're going to have still a primary oocyte, but we're going to have about 2 to 3 layers of granulosa cells. It's more than 2 layers, so it's granulosa cells now, not follicle cells. Then a vesicular or antral follicle, what we'll see is development of structures like the fluid-filled antrum. There'll also be a corona radiata and some other structures that are in this vesicular or antral, or sometimes called a tertiary follicle, and that's a mature follicle. Right now, though, it still contains a primary oocyte. When the vesicular or antral follicle develops further, it's essentially ready for ovulation. What's ovulated out of the vesicular or antral follicle is an oocyte that has just completed meiosis 1. So it's a secondary oocyte. It's a secondary oocyte arrested in metaphase 2. That's what's ovulated or bulges out of the ovary and is expelled into the peritoneal cavity and hopefully picked up by those fimbriae of the uterine tubes. What is left behind are those granulosa cells, those cells that degenerate slightly but change ultimately into a corpus luteum. And in the absence of fertilization, the ruptured follicle that becomes a corpus luteum would degenerate. But if there is fertilization of the ovum, then the ovum is going to require the progesterone and other hormones secreted or made by the corpus luteum until the placenta can take over. So these are the meiotic and follicular development events during oogenesis. So a really good image to come back and review. So let's compare now oogenesis and spermatogenesis. So first and foremost, we get 4 viable sperm from spermatogenesis. 4 spermatozoa that all work. Spermatogenesis has an error rate of about 3 or 4%. Let's compare that now to oogenesis. In oogenesis, we only got 1 viable gamete, 1 gamete that would work or could work if fertilized. And then we got 3 polar bodies. Polar bodies are going to degenerate. They're not viable gametes. So we get 4 viable sperm or gametes for spermatogenesis and just 1 for oogenesis. And there's a huge error rate for oogenesis. So betting it all on one cell, not always actually the best bet because this error rate is really high, but it's what oogenesis is. It's how oogenesis works. It's the physiology of the female reproductive system. So there's definitely unequal divisions when we're looking at oogenesis. And these unequal divisions ensure that the oocyte, though, has plenty of nutrients for its journey to the uterus. So it has to get from the fimbriae to the ampulla, all the way through the rest of the uterine tube into the uterus. That's about a 6 to 7 day journey, even after fertilization. So this unequal division, even though we have a high error rate, we're only getting 1 viable gamete, serves a purpose. Even though the polar bodies degenerate and die, we're giving this 1 viable gamete the best shot it has at making the journey through its uterine tube to the uterus. So make sure you know those differences, those kind of base differences between spermatogenesis and oogenesis, which are meiosis in the male and female reproductive systems, respectively. Next, we're going to look at the ovarian cycle. Now, the ovarian cycle is a monthly series of events associated with the maturation of an egg. There's 2 consecutive phases, and ovulation occurs mid-cycle. We're going to say, in general, a 28-day cycle is what we're assuming. So the first phase is the follicular phase, and it's named appropriately because this is the period of follicular growth where we go from primordial to primary to secondary to a tertiary follicle. It takes about 14 days in our 28-day cycle. At day 14, ovulation occurs, and then after that, it's the luteal phase, which is also appropriately named because after ovulation, what's left in the ovary, we're looking at the ovarian cycle, is the corpus luteum. So this is the period of corpus luteum activity. So for our 28-day cycle, this is day 14 to 28. So follicular phase first, luteal phase second. In the middle at day 14 or mid-cycle, ovulation occurs, which is ejection of the oocyte into the peritoneal cavity. So in reality, only about 10 to 15% of women have a 28-day cycle, and the follicular phase therefore varies. So that length of follicular growth can vary. But the luteal phase is always constant, so it's always 14 days from ovulation to the end of the cycle. So corpus luteum activity is constant. So let's talk about the follicular phase in more detail. We've talked about these follicles, but again, let's go into more depth. So the primordial follicle becomes a primary follicle. So what we saw there when we went back a couple images was the flat cells around a primary oocyte become cuboidal during that shift from primordial to primary follicle. And we're going to see the oocyte enlarge, but it's still a primary oocyte. The primary follicle then becomes a secondary follicle, and this is where we start to see granulosa cells, because we're going to get stratified epithelium now. We're going to get many layers of cells, semi-cuboidal, and they're going to form now around our primary oocyte. It's the granulosa cells and the oocyte that guide one another's development via gap junctions. So there's chemicals they use to communicate with each other. Gap junctions are communicating junctions. So let's look at these changes. So we have at number 1, really small follicles. I won't color them all in so we can see them, but they're small primary oocytes. These are the primary oocytes present at birth, and primordial follicles are also present at birth. So it's a primary oocyte wrapped in one layer of flat cells. So we even call them simple squamous, just for simplification reasons. That would be a primordial follicle. Next, we're going to see growth of the primary oocyte, and the cells start to take on a more, it's a horrible drawing, but a more cuboidal shape, but still one layer. So these are follicle cells. Then when we get a lot of layers, so 2 or more, so we can see many layers of nuclei here, those are granulosa cells. And our primary oocyte got bigger. And then we have a late secondary follicle. We're starting to see fluid-filled areas. So this is the beginning of the fluid-filled antrum. But this is still a secondary follicle because we don't have a true fluid-filled antrum, and it's just many layers of cells around it. And then finally, our mature vesicular follicle will have a big fluid-filled antrum, and we'll see structures like the corona radiata and the zona pellucida that are located in mature vesicular follicles. And it's the mature vesicular follicle that's carrying out meiosis 1, so it's ready to be ovulated. Now, it's the rupture of this follicle, the mature vesicular follicle, that causes ovulation. Now, even though at the beginning of the development of the mature vesicular follicle, it could still be a primary oocyte, by the time that ovulation occurs, the oocyte is a secondary oocyte. So it's an oocyte that has completed meiosis 1. This is a very dramatic looking event here, but this is ovulation. So we would have the ejection of the secondary oocyte into the peritoneal cavity. And this is what's left of the mature follicle, all of those granulosa cells. And this is the structure, these leftover granulosa cells, that becomes the corpus luteum. So it's formed from that ruptured follicle. And all this is occurring in our ovary. We can see many different states of development for the ovary. We have the primordial follicles up top near 2 and 1, and then we even have the antrum forming over here near 4. So use both images, the histographs and the schematic, to get a full kind of understanding of the ovarian cycle and what's occurring in the ovary during that time. So let's just take a closer look. Doesn't hurt to look at things twice. Primordial follicles present at birth, primary oocyte, surrounded by a layer of flat cells, those are follicle cells. Primary follicle, one layer of cuboidal cells, so those are follicle cells. And they're surrounding, again, primary oocyte. Secondary follicle, more than one layer of cells, so they're granulosa cells. They're usually 2 or 3 layers of cuboidal cells, and they're surrounding a primary oocyte. It's the secondary follicle that becomes the late secondary follicle. We do see that some connective tissue called the theca folliculi and granulosa cells cooperate and begin producing estrogens during this phase. So secondary follicle to late secondary follicle. It's the inner thecal cells that actually produce androgens in response to luteinizing hormone. So androgens like estrogen, for example. And that again is in response to a gonadotropin luteinizing hormone. And this is going to be the stage we start to see the zona pellucida form. It's much more visible in the late Graafian follicle, but the zona pellucida starts to form around the oocyte during the late secondary follicular stage. And then we see fluid start to accumulate between the granulosa cells. It's the beginning of the fluid-filled antrum. So that's what we see here, forming antrum, the theca folliculi, which are cells that are going to produce estrogens or androgens. It's the late secondary follicle that becomes the vesicular follicle. We get a full antrum. It's fluid-filled. It's expanding. It kind of isolates that oocyte over on one side in kind of a stalk of granulosa cells. And we'll see that the corona radiata is present. The vesicular follicular cells are going to bulge from the ovary surface. And we'll see that the primary oocyte completes meiosis 1 and becomes the secondary oocyte. And it's the secondary oocyte of the vesicular follicle that is ejected. Now keep in mind, this doesn't mean that all oocytes in a vesicular follicle are secondary, but by the time they're going to be ovulated, they are secondary oocytes. And so here's another look at our mature vesicular follicle, also called a tertiary follicle or a Graafian follicle. So ovulation happens mid-ovarian cycle. It's when the ovary wall ruptures. It's expelling a secondary oocyte. That's what's ovulated, a secondary oocyte that is haploid. And it's going to expel it with its corona radiata into the peritoneal cavity. There is a name for a twinge of pain that can be sometimes felt at ovulation. It's a funny name. It's Mittelschmerz. About 1 to 2% of ovulations release actually more than one secondary oocyte, which, if fertilized, would result in fraternal twins. Now, with identical twins, we're going to see the result of fertilization of one secondary oocyte, and then there's separation of that secondary oocyte that occurs, and that separation of daughter cells would produce identical twins. So here's another look at ovulation. Again, occurs mid-cycle of the ovarian cycle. And then the luteal phase of the ovarian cycle was the second phase. We just looked at all that follicular development, so now we're looking at the luteal phase, and this is the period of time during which the corpus luteum is active. The granulosa cells and the internal thecal cells that were producing estrogens and other androgens, they become the corpus luteum. The corpus luteum can also secrete progesterone and some estrogen, like I said, but this is what is formed of the ruptured follicle. It collapses, its antrum fills with clotted blood, called the corpus hemorrhagicum. But the luteal phase occurs second in the ovarian cycle, separated from the follicular phase by ovulation. And again, if there's no pregnancy, the corpus luteum degenerates. It becomes the corpus albicans, or a scar on the ovary, in about 10 days. But if pregnancy occurs, the corpus luteum produces hormones that sustain pregnancy, mainly progesterone, until the placenta takes over at about 3 months. And just another look at the corpus luteum, which is what the ruptured follicle or the remaining granulosa cells become. It's a hormone-secreting structure. And just an overview, I highly recommend spend some time on the schematic and microscopic view of the ovarian cycle. Be familiar with the ovarian cycle. It's 2 phases, follicular and luteal, separated by ovulation. And that's it for Part B of the reproductive system.